Assalamu alaikum. I hope you all are good. I is Shamansoor, roll number 20, Department BS Biotechnology. Today my presentation topic is test for anti-aging creams. So first of all, what are anti-aging creams? Anti-aging creams are moisturizer-based skin care products marketed with the promise of making the consumer look younger by reducing, masking, or preventing signs of skin aging. Secondly, many anti-aging products and treatments have not been proved to give lasting or major positive effects. It reduces wrinkles by less than 10% over 12 weeks, which is not noticeable to the human eye. Another study found that cheap moisturizers were as effective as high-priced anti-wrinkle creams. However, recent studies at Manchester University showed that some ingredients have an effect. So moving on, ingredients which are used in anti-aging creams. It may include moisturizing ingredient. First of all, retinoids. It is used for instance in the form of retinol palmitate, which reduces fine lines and pores. Second one is epidermal growth factor. It stimulates cell renewal and collagen production in the skin and strengthens elasticity and structure. It reduces fine lines, wrinkles, and sagging. It heals wounds and burns. Alpha hydroxy acids and beta hydroxy acids are other chemical peels. These help to dissolve the intracellular. First one is glue that holds dead cells together on the skin, enhances the exfoliation of the epidermis. It causes skin infections maybe. Peptides such as acetyl hexapeptide 3 and copper peptides, coenzyme Q10, antioxidants are substances that may protect cells from the damage caused by unstable molecules known as free radicals. Sunscreens provide a high level of UVA protection against the effects of UV radiations such as wrinkles. And the last one is vitamin C, which is a very important ingredient. So moving on the testings of anti-aging creams. First one is microbiological testing. It includes product samples are tested using different methods highlighting the presence of bacteria, fungi, or yeast. The sample is also submitted to a challenge test, also known as a preservative effectiveness test (PET), in order to identify the risk of harmful microorganisms growth. It contains different microorganisms that can be harmful to consumers during its use. Using a product, customers can bring bacteria that can mix with other chemicals. The microbiological testing allows manufacturers to ensure that it is free from any harmful microorganisms growth. Product samples are tested using different methods highlighting the presence of bacteria, fungi, or yeast. The sample is also submitted to a challenge test, also known as preservative effectiveness test, known as PET, in order to identify the risk of harmful microorganisms growth. The second one is stability testing. Depending on its environment conditions, cosmetic product can be altered and become unsafe for consumers. During its shelf life and consumer use, the product maintains its function, physical aspect, and its chemical and microbiological quality. The product samples are put under real conditions to determine its stability and physical integrity. No color, odor, or any physical aspect change. The stability test is required to complete the PIF for product sold in the EU. Third one, which is very important, that is safety and toxicological testing. In order to make sure the raw materials and finished product do not present any risk for consumers, manufacturers must do a safety testing that includes a toxicology test. It allows them to determine if any substances of the product and their mixtures present a risk when used by customers. Several tests are included to highlight the product effect when in contact with skin and eye: skin irritation, corrosion, penetration and sensitization, eye corrosion and irritation. Moving on, performance testing. Performance testing is made to demonstrate product claims and ensure that they are real. It tests the product functionality, usability, durability, and performance. It is related to advertising and packaging, as everything you will be promoting must be proven. For example, if you advertise that your hairspray will provide volume to the consumers, you must do an adopted test to prove it. Fifth one, compatibility testing with packaging. The packaging should also be tested, and especially when in contact with the finished product. Chemicals can easily react with other substances or materials, and that could present a risk for consumers. Compatibility test will check if there are any corrosive effects between product formulation and packaging, any alterations on product function, or any alterations on product aesthetic as a result of contact with the packaging materials. Many other tests can be made in order to ensure product safety and give more transparency to consumers. All the test records must be gathered and stored carefully, as they will be requested if your facility is inspected. LASCOM has developed a feature that will help you to gather and store quality documents analyses in your PLM solution. Laboratories are able to enter information directly on the platform, allowing you to save time and ensure data accuracy. Biopharmaceuticals. The exposure to the ultraviolet radiations is the key source of skin sunburn. It may produce harmful entities, reactive oxygen species, leading to aging. The skin can be treated and protected from the injurious effects of ROS by using various pharmaceutical formulating such as creams. Cream can be loaded with antioxidants to quench ROS, leading to photoprotective effects. Moreover, modern medicines depend on protection or treatment of human diseases. These formulations may serve as a cosmeceuticals to protect skin against injurious effects of UVR. The botanical studied for the dermatological use in cream form. The observed anti-aging effects of cream formulations could be an outcome of a coordinating action of multiple constituents of numerous botanicals. The phenolic acids and flavonoids appear effective against UVR. The second important thing is cytotoxicity testing on the cultured human skin fibroblast of the semi-purified rice bran extracts by the SRBS. The semi-purified rice bran extracts containing bioactive compounds FO and P were tested for cytotoxicity by the SRBSA in a 96-well plate. The 1x104 human skin fibroblasts were added in the total volume of 200 into each well in the 96-well plate and left for cell attainment on the plate for 24 hours in a 5% CO2 incubator. Cells were then treated with 20 of the 5 serial concentrations of the extracts from 0.00 to 10 mg/ml and kept at the same condition for 24 hours. After incubation, the cells were fixed with 50% TCA at 4 to -1 degree Celsius for 1 hour and washed with distilled water and air dried overnight. The fixed cells were then dyed with 100 of 0.4 SRB solution at room temperature for 30 minutes. After the dye was removed, the cells were washed with 1% acetic acid and air dried overnight. The percentages of cell growth were calculated by the following equation, as we can see at the last. Preparation of gel and cream incorporated with the NIOFOP. Gel preparation generally stirred by magnetic stirrer until homogeneity. The mixture of mineral oil and its concentration. The pH of the gel was vigorously mixing until the homogeneous gel was obtained. For cream preparation, theoretical emulsions are formed by heating oil and aqueous phase separately to about 65 degrees Celsius and subsequent mixing together. Therefore, in this study, the oil phase and aqueous phase, which contains nizomes, were heated. The oil phase was then poured into the aqueous phase and stirred until the mixture was cooled to 40 degrees Celsius. Mixed and continuously stirred until the emulsion was completed. Cream RBO was prepared by the same procedure, rice bran extract in the aqueous phase. The amount of rice bran extract in cream RBO is equal to those in cream NIO. The skin anti-aging effects of the combination of the unentrapped and entrapped rice bran bioactive compound in cream RBO was investigated in comparing to that of the entrapped rice bran bioactive compounds in cream NIO. So that's all from my side. Thank you. Allah Hafiz.